Thank you very much for attending the webinar this afternoon, or this morning if you're over on the West Coast. So a little bit of background on Horizon Discovery before I jump into the details of the talk. So Horizon Discovery, we're a genome translational company where we can offer services from cell line engineering to developing tumor microenvironment assays with our CRO products. And what I'm going to talk about and focus on is the Horizon Diagnostics division that focuses on reference standards, and in particular this one will be discussing multiplex reference standards and their use in next generation sequencing platforms and similar platforms. Now, one question that we've always asked ourselves in the lab and have started posing the question to other labs has been, how do you know how well, or how do you know how well your molecular assay is working today? And we've kind of started to change that question a little bit more and really push people and asking, do you assume that your molecular assay is working today? And when you push that question, we often get the answer, well, the positive control that you're running gives you the expected result, and the negative control that you're running gives you the expected result. And so following on from that, we can, the next questions are really, what are the controls that you're using, where are they from, and what is their suitability and how they validated. The negative control can often be, in some of these kits, simply water, and the positive control could be a short oligonucleotide that has no relevance or no genome relevance in terms of comparisons to reference serial patient samples. And our understanding in our work with the key proficiency schemes, both in Europe and in the US, has been that a lot of laboratories will place a lot of confidence in their controls or reference materials without really understanding how well they work and how well they've been characterized before they get into their hands, essentially. And these are the questions we are continuing to ask, and we're sort of, hopefully I'll be able to present some data to show how we're trying to address variability in molecular assays. What I'm going to do first in the talk is focus on where variability can sneak into assays, and then we'll have a break. There's a short break in sort of half time, as it were, and then after that I'll really delve into the details and the data that we've got in terms of our multiplex reference standards and testing and applying these standards on Ion Torrent, Mi-Seq platforms. So if we focus on this slide, it's a very simple schematic. You get a tumor sample. It may go off to histology and go through IHC or even cytogenetics lab and be tested for FISH. What we're focusing on really is if it goes towards a molecular pathology lab or molecular genetics lab, where you'll have DNA extraction, it'll be genotyped, and then the genotyping will, together with other factors, govern the diagnosis of the particular sample and patient, and then direct a therapy. All the way through this process, there is opportunities for error or variability to sneak in: sample quality, sample heterogeneity, DNA quality from the DNA extraction, platform variability, and mutation characteristics. I'm just going to tease each one apart in the next few slides. So as you're sort of, another way of summarizing the last slide is the diagnostic paradigm, where you have genomics, transcriptomics, and proteomics. You can be looking at the DNA or RNA. You can be looking at DNA using FISH, or you're looking at DNA or RNA using genotyping, or focusing on the proteins with IHC. If we're looking at DNA, and that's what the focus of this talk is, we're looking at genotyping particularly, and historically it's been looking at SNPs of rare mutations, small deletions. But I think one thing that next generation sequencing is opening up is the opportunity to look at more complicated gene rearrangements, and certainly there's been progress taking on and identifying the different EML4 ALK translocations and rearrangements using NGS, and that's actually, as a method, could replace FISH for testing of email for ALK or even IHC, where there's some movement in that area too. So sample heterogeneity. All human tumours will display heterogeneity, and this is driven either morphologically or epigenetically. And there'll be a lot of genetic diversions within tumour cell clones. And this poses a big challenge for any molecular diagnostics, and it can sort of be exacerbated with next generation sequencing. So if you've got say a wild type sample all the way through to a patient that has, if you've taken out a tumour and you've got 100% tumour cells, you'd expect you'd be able to detect this with Sanger sequencing with standard PCR methods. The more challenging is when you're down to patient samples where you may only have 1% of the mutation present, or maybe 10% of the mutation present. If you only have, say, 1% of the mutation present at the phenotype level, at the genotype level, it could be half, because it might be heterozygous mutant cell clones you're looking at, and this will govern whether or not you can or can't actually detect the mutation. Now, when you're applying this to multiplex assays, and I've kind of put together some very simple schematics to try and sort of help describe what I'll try and say, is if you've got a multiplex assay, so NGS, where you're looking at, say, 40 or 50 different genes, you need to have confidence that if a sample comes in, say sample 1 comes in and has a mutation in gene X, you have to be confident that you'll be able to detect that mutation. So if it's a 10% sample, can your multiplex assay detect that 10%? If the second sample comes in and it's a very low allele burden and it's on gene Z, can you detect gene Z at 1%? And I mean this is hypothetical, but if there was a patient sample that came in with multiple mutations within one sample, would you be able to detect the different mutations as low as 1%? Why this matters is that if you look at and this is a summary of the panels that are available currently, you've got a variation of how many genes it covers, so from as low as 46 up to 146. And when you're first setting up the assay and also checking the assay works on a daily basis, how do you know that you're able to detect mutations within these genes? And if you sort of question how you're able to detect it, what's the limit of detection for each of those genes? And I think this is something that people have started to ask a lot, and they've asked us, and this is where we've come in to help in a way to answer these questions, because if you're able to detect, say, KRAS G13D at 1%, that doesn't necessarily mean that you can then also detect BRAF G13D at 1%. And so you do, if you're going to have a panel where you're going to be testing for 46 genes or 142 genes, you need to be able to do essentially a limit detection test per gene and almost per mutation as well. So even with BRAF, you could envisage that you need to go into V600E, V600K, potentially V600D or R. And likewise with KRAS, you've got the codon 12 mutations, codon 13, codon 61, codon 146. And it brings on the question at the bottom of how do you go about validating the complete panel. Now, one thing that's come up with our work with the key and leading proficiency schemes in Europe and in the US is something that was, I think, sometimes forgotten, and that's the quantity and concentration of the DNA that's used to run on molecular assays. So it's well known that typically molecular pathologists we don't have any control over formalin fixation for some of our samples. So if we're unlucky, a sample could be taken on a Friday afternoon, put into formalin, and then be fixed over the Friday afternoon, over the whole weekend, before it then arrives ready for us to process on the Monday. And so in the worst case scenario, a sample could be fixed for 72 hours Alternatively, if it's caught just before we go along to the lab to pick it up, it could be just 1 hour in formalin. So, and obviously formalin fixation will govern the quality and yield that of DNA you get from samples, as well as other things like the size of the sample and such like. There's then different DNA extraction methods. Some will be good at giving total yield, but the quality may not be as good. Some will be good at giving you very high quality, but in doing that, there's a concession that you get a lower yield or a lower concentration. Some may be there to give you as high concentration as possible, but then the caveat is you have a lower yield. It's always a balancing act. What's key is how much DNA goes onto the platform and how you quantify the amount of DNA before you put it onto the platform. So each platform needs different amounts of DNA, and that's the nature of the beast. Whichever platform you have, you'll work towards being able to always have sufficient material to get onto the platform. What's critical though is once you extract the DNA from your sample, that you actually use the method for quantification that's going to give you as accurate a result as possible. And we know, and we've got a lot of data internally that indicates that nanodrop will, if you've got a concentration less than or around 10 nanograms per microliter, it will always overestimate the amount of DNA you have. And so this is an example where this is an example from our lab where we've done extraction method from FFP material. If we take and I'll start with the nanodrop first, because that was what naturally we do that first because it's a lot faster. We did a nanodrop measurement, and the measurement of DNA from the material was 6 nanograms per microliter. The next step was we were doing genotyping, so we needed 20 nanograms to go into the assay. If we were taking the nanodrop reading, this would then dictate that we needed 3.3 microliters. Now, on the other side of things, we then take this through to picodegreen, or it's similar to Quantifloor as well, which is a sister dye. And measuring with picodegreen, we've actually only got 2.5 nanograms per microliter. And if you've got, again, if you've got 20 nanograms required for genotyping, this would mean we need 8 microliters to achieve that. Now, obviously, it's from one experiment you can't say which one's correct, but through the DNA quantification validation and comparing these two assays to a digital PCR assay, as well as doing various dilutions from a high concentration, say 50 nanograms per microliter, which is accurately read by both all the way down, we have full confidence that this picodegreen is very accurate below 10 nanograms per microliter. And so if we'd only measured with nanodrop and taken this 3.3 microliters, for this particular assay, we would actually be running almost somewhere between 2 and 3 times less DNA than is required. And if you've got, it won't necessarily make an impact if you have, a bucket loads of mutations. If you've got an allelic frequency of 50% in your sample, probably won't make a difference. But if you're on the boundary of your limit detection, so if you've got somewhere between 1% and 5% tumor sample, and then you don't put in enough DNA, you're then putting in, you're not going to have enough copies of the mutation to actually be able to detect it with your assay. And so it's absolutely critical that this step is addressed, and it's something that's certainly vital for any of the NGS, for preloading onto NGS platforms. So the next step is, or the next point to at least look at is the different levels of sensitivities that you can detect with different methods. Everyone knows, I think it's generally accepted that Sanger can be anywhere from 15 to 25%. We've got PyroSeekers and Roche and Sequenom systems, maybe around the 5%. There's a number of homebrew methods that was been interested in Vienna labs, around the 1%. And then everyone's kind of saying that NGS and multiplex assays is going to be leading the way, and we'll be down at this area of, at least 1%, maybe even lower than 1%. And I think it's certainly true, as long as you're controlling for each mutation that you're looking at. And I'll show data in the second half of the talk to kind of show where we're definitely in that area and where somewhere we're in some cases we're falling short. What's interesting is, and this is data from Sanger sequencing, but it will be true, and the data I'll show later will also highlight this is true for NGS as well, where different mutations will give you completely different limit detections, even if you're using the same method with similar, even if you've got the same kind of algorithms for designing your primers and probes. Here it's very relatively simple because it's Sanger sequencing, but with Sanger sequencing, this lab was able to get 5% for G12A, but then could only detect PI2 kinase E545K at 25% as best. And in a way, it's the multiplex assays or platforms. You could say they're as good as their lowest denominator. It depends. If you know that your assay can detect KRAS at 5%, but you'll never be able to detect 1%, I think that's okay. But it's important you know what the lowest is per gene or per mutation. So the materials out there at the moment, I think this has been sort of before Horizon Diagnostics came along, is you could use synthetic oligos as reference material, cell line mixtures, and primary tissue samples. In the NGS field, cell line mixtures have been used a lot, and I think there's a lot of very useful data and very good data indicate that can be used as reference material. What the big drawback, or the one big drawback with the cell line mixtures at this point, is because they've all been validated and verified using NGS, you're kind of validating the control that's controlling the system, which inherently is going to at some point fall down. So because if you think about validating material that has an issue, you'll validate it and say yes, it's okay, and then when you're testing for it, your system is okay, but it has that issue, it has that problem that may be inherent in that particular cell line. With going ahead with cell lines, and this is an example I've sort of shown when I've presented about singleplex, there's a question that's just popped up. Oh no, I'll carry on. So one thing that we looked at, and we're working with a collaborator, is and this is actually a proficiency scheme, so they were doing a proficiency scheme and ring trials, so they were sending samples out to a number of labs, and they actually found that the labs were able to detect mutations down to 1% with Sanger sequencing, and this obviously is just not really. It was an unusual result. And what we did was we took their samples and we ran their samples through our digital PCR platform, and where they generated a detection curve from 100% all the way down to 1%, we tested that and we found that at the lower end, so the 1% was actually a 7% allele burden of the EGFR deletion, the 2% was actually 14%, and so on. And so where the Sanger sequencing guides could detect what they thought was 2%, they were actually detecting 14%. When we delved into it further, it came down to the fact that there was a massive copy number amplification in the cell line they were using. So they were using this cell line that had a copy number of 15. And what was interesting is we sourced an independent cell line from A2CC, and we did a copy number analysis of that. It came out at 38. And so although they actually got their cell line from A2CC as well. So although they got a cell line from A2CC and we got a cell line from A2CC, for this particular cell line, the difference in copy number for EGFR was over twofold. And what's also something we're always aware of, especially when we get cell lines in from external sources, is they're often heterogeneous. And so even looking at HeLa, there's a population of cells with a 2N EGFR as well as population 3N EGFR. So it's important that if you are using cell lines, that they are fully characterized before proceeding with using them as reference material. So that kind of brings me on to kind of the final slide of this half of the talk, and I thought it was good to define what a reference standard is, in our eyes at least. So we see it as an independent or external validated reference tool. It will allow you to understand the sensitivity and limit detection of your assay. And it will also allow you to monitor the reproducibility of your assay. In terms of development and validation, it's absolutely critical. But I think what's becoming very apparent is that it can be used, and particularly for NGS, it can be used to tell you if your assay is working today and there hasn't been any bias or any sort of misreads within a sequencing run. So let's get back into the second half, and this is for me, this is the more interesting part because it's actually got some more nitty-gritty of the data. So it's important for us or me to introduce how we make reference standards to then tell you how we can test the NGS platforms. So we take wild-type cell lines, normally from ATCC. We do a single cell dilution to then ensure we've got a clonal wild-type cell line. So if we saw the previous example, we'll know that if you look at EGFR at any of those cells, it will have a copy number of 2, and there won't be any heterogeneous populations within that. We then, once we have that cell line, we will then validate it, and I'll go into more detail on the next slides using various methods. After validation, we will then use our gene editing technology, known as Genesis, and Genesis will then, we're able with Genesis to incorporate a number of different mutations or different types of mutations into any cell line. So with Genesis, we can incorporate SNPs, we can do short deletions, large deletions, we can create translocations. And the key to it is that all of these mutations have to occur at the endogenous site, and will then generate essentially a mutant cell, which will then do a further single cell dilution to ensure we then have a clonal mutant cell line. The cell line validation that we do, we'll do SNP6 on the original parental cell line to ensure that if we're starting with an HCT116 cell line, it is an HCT116 cell line. We'll then confirm that the integration of the mutant is at the endogenous site. We'll look at the expression either by standard cDNA PCR. We've now actually moved more into looking at it at the droplet digital PCR level. We'll confirm the clonality and gene copy number with predominantly digital PCR. And the outcome of this is that we have the original wild-type cell line and then the engineered mutant cell line. And these two cell lines are essentially identical except for that one mutation. And so they are, in terms of patient genetics, as close to patient genetics as you can get in cell lines. We've made over 400 or somewhere between 400 and 500 cell lines as part of Horizon Discovery, and within Horizon Diagnostics we have, I think it's something like 45 cell lines that are oncology specific and cover the key genes like BRAF, EGFR, KRAS, NRAS, and we've taken these forward into generating reference standards. And what we've tried to do is tick all the boxes essentially for what a reference standard should be. So we're able to define the copy number, we're able to define the allele burden. It shows genome heterogeneity because it is, any of the SNPs are incorporated within the endogenous site. We can develop these, take these cell lines and provide them either as DNA or FFPE, so have compromised DNA. They will show tumor cell genetics because you have the wild type or the mutant, and they're identical except for that one mutation. And we will validate them with a non-NGS platform, which means that when you apply it to NGS, you know that it's been externally validated on a platform and on a system that is independent of what you're actually running. So this is where it gets good in my eyes. So we've taken a number of different cell lines, extracted the DNA, and then mixed them all together to create precise DNA dilutions, and then analyze that with digital PCR to generate what we've termed the quantitative multiplex DNA reference standard. Before I show you any of the data, I wanted to kind of really give you confidence that what we've generated is a standard that we believe, we trust in, and we have full confidence in. So for us to make the multiplex, we need to first validate the digital PCR platform, and to do that we took the singleplex genes first. So for example, we took a mutant, and this example is KRAS G12D. We took KRAS G12D, extracted the DNA, we took the corresponding wild type, extracted the DNA, and then we created a dilution curve from 50% all the way down to 0.1% allele burden. And this is the digital PCR data, and it's true for all of the assays that we've performed for generating the quantitative multiplex assay, where you have the predicted value, so you've got 50% all the way down to 0.1%, hitting pretty much dead on the actual called by the system, and you know, perfect linear regression all the way down to 0.1%. We rolled that out for all the mutations. Obviously, we didn't want to be in the position where we've got the chicken and egg, where we're using our standards to validate the machine, that we're then using our standards to generate additional standards. So we took our standards and put them on external platforms. So this is actually FFPE material. So we grew up our BRAF mutant and we grew up a BRAF mutation, and also the wild type mutation, and we mixed them together to generate a allele frequency of 25%. And then we then fixed in formalin, embedded in paraffin, and then sectioned, did the DNA extraction, and then ran it on COBAS. And we had the 25% blocks we created came out dead on 25%. We then had a 6% block and a 3.5% block, and the 6% block comes out here, and then the 3.5% block comes out here. And so it gives us, you know, the external validation gives us the confidence knowing that it's confirming both, it's confirming what we're seeing at the digital level, and also confirms the fact that, you know, because we're starting from an engineering point of view, we kind of know, we already know that 50% is a 50%, and when you dilute it, it's doing what it's expecting to do. We then decided to go, you know, one more step further, and so we did analysis on the genome analyzer, and again generated dilutions from 50% down to 0.5%. And this is actually it's comparing runs. This is run A versus run B, and where the dilutions, where you had the dilutions come out on run run A and run B, again matched each other and gave, you know, it gave us the confidence to proceed with the quantitative multiplex. So this is what we did. We tried, so we mixed I think it was 8 or 9 different cell lines with the different mutations. So we're covering BRAF, KITs, EGFRs, 4 mutations, KRAS, NRAS, PI3 kinase. This is the predicted frequency based on the cell lines that we have, and mixed by mixing them together, we'd expect to come in at 10%. And when we hit 10%, what's really striking is that we hit what we're expecting, plus or minus 1%. And so you've got, 10 came out at 10.5. There's a 0.5% out there, and 1% there, 1% there. And so we were, you know, very pleased with what we were able to do. And, you know, that was nice. But what was even nicer was then when we started to apply it to non-digital PCR platforms. So we put it onto using the KLOS target rich panel on the Mi-Seq. 10.5 comes out 10.5, 10 all the way down. There's a few variations, so the EGFR G71S comes out a bit lower than expected. But then what kind of struck us, and you know, this in a way it was kind of like needed more investigating, was there was two mutations that just fell out. And it wasn't, so you've got E545K at 9%, it wasn't detected on this panel. Likewise, the NRAS Q61K again wasn't detected, and that's at 12.5%. So it's strange that you can detect mutations at, like, say, 2% for the EGFR, that couldn't detect down here. You know, we're investigating why that could be, but essentially it's, you know, it's a panel fail for those mutations. And you know, if this was at some point being applied clinically, you'll be at a point where, you know, you could be missing NRAS codon 61s if you're using this particular panel or this setup. We then rolled this out. That was on Mi-Seq. We then rolled this out to 3 different partners where we, using slightly different panels on the Iron Torrent. This is what we see with the digital PCR platform. In the majority of cases, the allelic frequency comes out as expected or close to as expected. For this one, is an interesting kind of looks like an allele bias where maybe one strand is read over the other strand because you don't actually get the 9% as expected, whereas the other 2 panels you do. Again, I think what flagged up, and for us it was a worry for these systems, was the EGFR deletion. Again, so it is a low allele burden, so it is 2%, but it wasn't detected by these 2 partners, but it was detected by this. Likewise, the T790M not detected on using the hotspot panels, and also we have this, and you know, this is, you could say this is similar because they're using this, you know, the same panels, but then, you know, this for some reason G12D just fell out of this result, but not of the other 2. So it kind of, you know, we're working with more partners to really try and understand where the variability is or why mutations might fall out. I think what it illustrates is that whilst as we're moving into the age where we can do multiplex assays, we really need to have normalized standards to ensure that you can detect when mutations do fall out in a run, so you can then try and work out why they're falling out, or you know, where did the data go, or why wasn't it supported in that particular run. I mean, overall, I think the data's, you know, it's relatively reassuring. So you've got, you know, this lab can detect, you know, every single mutation we threw at them. This one is, you could say, you know, it's hitting 8 out of 11. This one's hitting 10 out of 11. And as long as they know that they're hitting so that's 9 out of 11. As long as they know that they're hitting 9 out of 11, they just have to be aware that those 2 are they're not optimized assays for them. And it could just be that they might be able to detect it if it was 5% or if it was, say, 10%. And that's something I'll talk about in the next few slides. I think from where we are and talking with a number of our partners as well as the quality assurance schemes, is with any of these multiplex and NGS platforms that you need a normalized reference standard to ensure everything is working as it should do. Now we thought we thought that 11 mutations was good, but not enough. And so what we did next was we looked at known SNPs that were endogenous to the cell lines, and then we developed digital PCR assays against them, and then we went in and tested to see if the predictive frequency based on the mix in the cells actually hit the actual frequency. And again, if you look at all the numbers, you know, they're plus or minus about 1%. It might be a bit of a small outlier there. And so we're now in a position where we have a quantitative molecular multiplex reference standard that has essentially 30 mutations, which range from rare mutation SNPs to the deletion. We've got frame shifts. We've got 5' UTRs in there, and you can essentially look at. I think it covers almost every chromosome as well. I haven't got the chromosome details on there, but I think these will cover the majority of the chromosomes as well. So you can actually look at if there's any amplification bias between chromosomes. And we're now at the point where we can reanalyze some of the Iron Torrent and Mi-Seq data to see how well the original runs work to pick up these different SNPs. And what's nice is we've expanded this. So we have this available as DNA, which in a way for us is now relatively easy for us to mix and generate. We also have it as FFPs. This is where we'll grow up the the 8 or 9 cell lines. We'll mix those cell lines together in the defined dilutions. We'll then fix them with formalin and embed them in paraffin and then create sections. So you can then really test the whole process from start to finish, and you can really test how well the DNA you can do the DNA extraction and then monitor the yield and then load on essentially compromised FFP extracted DNA onto your platform. We also looked at different ways we could actually start to answer some questions that came up in the first study, particularly around the deletion in the T790M, the way they fell out. And what we've done is, so the way that we engineer our cell lines is that we will typically take a cell line background, so for example RKO, that's relevant for the mutation. So for all of our EGFR mutations, we've engineered them in an RKO background, which means that we have 4 cell lines that are identical except for these mutations, and we also have the wild type that's identical to these except for mutations. And so what we've done is we've generated a gene-specific multiplex reference standards. So here we have it's focused on EGFR, and then we also generated another one that is focused on KRAS, and what we've done is we've focused on codons 12, 13, and 146. And because of the nature of our cell lines, there are a few additional endogenous mutations that come along for the ride, and they're actually can be very useful mutations to then use to monitor inter-variation. So because we're able to, like I said, these are all in the same background, it means you have a starting material of, if you've got starting frequency of 12.5%, you can then start adding wild type to then dilute the mutations down, and because it's the same wild type, they will dilute exactly the same. So you can create a standard that's 10%, you can then create a standard that's 5%, you can then create a standard that's 1%, and so you can then really monitor to see where a particular mutation may fall out, and you may be able to detect the deletion here, and then the 858R and the 861Q down at 1%, and then the T790 might be at 5%, and the same would be true for the KRAS. And then in having the endogenous additional mutations, it means you can actually just measure that, you'd expect these to always be the same per assay. So as you did the dilutions, you'd expect these to always come out at 50%, so you can then monitor the intervariability. Because we've generated all these different mutations, we've now characterized the SNPs within a number of the wild type backgrounds as well as mutant backgrounds. We can really start to customize reference standard per assay, and it's just, we've got all of these mutants. You can choose the genes, choose the mutations, choose the allele frequency within reason, and then pick whether or not it would be useful as DNA or FFPE. It'd be nice to say that we can do that do this for every single customer, but obviously, the workflow to generate all these is tricky. I mean, we've got to the point where we have the original multiplex standard where it has 30 mutations covered. We've now got these 2 new ones. For me, I probably should have introduced myself a bit better at the start. I'm the product manager, so I'm essentially responsible for developing all of these standards and manufacturing them. And so it's always good for me to get feedback from you and from your colleagues to really understand which mutations we should be focusing or which allele frequency we should be aiming for. And although we may not be able to make the custom standard for every single lab, we can certainly generate a custom standard that will be able to serve 90% of a lab's needs, and then we can have flavor 4, flavor 5, flavor 6, and then just build on it based on your feedback. So if you've got any ideas of multiplex standards that you require, and maybe it's in genes that we haven't got already, because we can engineer them. If there's genes that we're missing, then we can go back very much further up the drawing board and engineer cell lines that can then feed into this pipeline. Sort of coming to the close of the webinar, we are now working, or we have been working very closely with all the proficiency schemes, and they have really helped us to push us in the direction of what is needed. Like I said, they help to show which standards are required, and particularly in the NGS. I mean, this is a very new field, and I don't believe there's possibly 1 or 2 proficiency calls going out this year, but there's not that many because it's relatively novel. And it brings us onto why we're doing what we're doing. And we really believe that, you know, as molecular diagnostics does evolve, it's critical that we're able to deliver to the patients as good a diagnostic test as possible. And we started with questions. I started the talk with questions, and it is questions that I like to end in as well. When you're looking at the diagnostic tests that we're doing, whether it is singleplexing, whether it is multiplexing, whether it is NGS, is what does a diagnostic error mean to the patient? It will vary on the different genes that you're testing. What we want to eliminate is as much variability as we can to achieve better outcomes. And the way that we are trying to do that is by developing these reference standards that are consistent, well characterized, and because we're generating them in cell lines, they're evergreen, and so we can continue to generate FFPE blocks or DNA as needed. I think one of the things that's quite striking, certainly in Europe, is that there's in some of the proficiency schemes that have been run, there's maybe a 5% error rate on some of these tests, which in some ways is a vast improvement on where it was maybe 2 or 3 years ago. But 5% is essentially 1 person in 20 would get the wrong result from a diagnostic test, and 1 in 20 is kind of probably one that would be someone that you would know, because if you were to go to the numbers. And I think for me personally, that's not good enough, especially with the technology and especially with the advances we have, that diagnostic tests should be in the 1% or lower in terms of a rate, or if we look on the positive side, 99% better or higher. So I think I'll finish on that. Thank you very much for attending the webinar.